We got a great team from Diagnosis, and we also have guest speakers tonight. Tomas Aleman from the Shea Eye Institute and David Birch from the Retina Foundation of the Southwest. Both of them have used the FST very extensively over the past several years and have published quite a lot on it. And so they're here to talk about that today. So Jeff, if you want to take it from there.
I will. Thank you, Natalie. I've got a couple of charts. Hello, everyone. I'm Jeff Farmer, CEO of Diagnosis. I have a couple of charts to get us warmed up before I turn things over to our keynote speakers, Dr. Aleman and Dr. Birch. So I will get us started. A couple of introduction charts for those of you who do not know Diagnosis. We were founded in 1984. We have offices just outside of Boston in the United States, Cambridge in the UK, and also in Dublin, Ireland. And we have distributors around the world, including most of the major countries in Asia, Australia, South America, and other parts of the world. Our systems that we sell today and have been developing for quite a few years span a number of categories of diagnostic tests. This chart is just a brief introduction to that. We have Sweet VEP, which is an objective test of vision, visual acuity specifically. Today we'll be talking about FST and dark adaptometry, so that's a subjective test of function, but it is quantitative, as well as electrophysiology, a full suite of ERG, VEP techniques that can be run on our systems, and pupillometry. Our systems come in two primary platforms, a tabletop system that is called the E3 desktop system, as well as a cart-based system, which is the profile system. Both of these systems offer the full set of stimulators, test modules, protocols. They're just in different form factors. The test capabilities on these platforms include all of the standard ICEV protocols, extended protocols from ICEV, and many dozens of other custom ERG and VEP protocols. They also are able to be upgraded with modules including the DiagnosysFST and dark adaptometry. We'll talk a lot about DiagnosysFST today, as well as Sweet VEP and pupillometry. And all of these tests on both systems run on one software package we call SBION. So a quick introduction to functional visual psychophysical diagnostics. It's a suite of diagnostic tests that's used for a broad array of patient types, including low vision patients. This could be in a clinical trial, drug discovery, delivery diagnostics, including gene therapies and other purposes. Clinically, these techniques are starting to be used for identification and monitoring of inherited retinal diseases, as well as glaucoma in other patients where the disease is in its later stage. FST can be quite useful where visual field leaves off, and our guest speakers today will talk a little bit about that. AMD patients, dark adaptometry in the last few years has started to be used for the identification of early macular dysfunction, and then other effects such as traumatic brain injury as well. In both cases, for the DiagnosysFST, as well as dark adaptometry in general, the technique for the test is a brief flash of light is presented to the subject on one of our stimulators, and then the subject responds using a button box as to whether or not they've seen a flash of light. In the case of FST, it's a forced choice 2-button yes or no response, and in the case of dark adaptometry, typically it's a single button box. These tests run on both our color dome as well as our color flash stimulators and are offered on both the E3 and profile systems. So basics of dark adaptometry. On the left-hand side of this chart is what we would call a full test, where you measure rod and cone during the test itself. This chart, if you've never seen this before, you read from left to right, and as you go down, this indicates better sensitivity to lower flashes of light. So this test, a bleach of a very strong background light was shown to the subject for a few minutes, and when that light was turned off, the test began. Sensitivity to red and green was tested in this test, and in this case, you can see a cone plateau, which is the first plateau here. That's the cone sensitivity to red and green. Then as the patient further dark adapted, you can see plateaus that happen in this part of the chart for red and green, and that's the rod sensitivity to red in one case and green in the other. And as we talk about FST, FST is basically a very clever test that was developed a few years ago to measure those final sensitivities after dark adapting. So what is a DiagnosysFST full field stimulus threshold test? This test measures the sensitivity of the entire visual field by testing for the lowest luminance, which elicits a visual sensation perceived by the subject. As I mentioned earlier, it is subjective. We need the patient response during the test, but it is very quantified. We can measure quality measures of the test itself, and this tests the basic vision of a subject. We use proprietary methods within our software to calculate the threshold based on patient responses during the test. The basic test process, as I mentioned earlier, there's flashes of varying luminance are presented. These are paired during the test with an audible beep. So when a beep happens, the patient is asked to respond. If they saw a flash of light, they would push the yes button, and if they don't believe they saw a flash of light, they would press the no button. Based on the response that our software receives then from the preceding flash and tone that was given, our software works its way through a set of automated proprietary algorithms until metrics are met, the test is ended, and a threshold is reported. There's a few different options for the test. Dark adapted threshold tests traditionally are the types of tests that have been done on our FST systems. This year, we've released a light adapted set of FST tests, and both Dr. Aleman and Dr. Birch have started testing those in their clinics this year. We also have the capability to do subfield stimuli using an eye mask that plugs into the color dome. And then in the color dome itself, which traditionally has been the stimulator people have used to do DiagnosysFST, we have a very wide range of flashes from well below the scotopic threshold of a normal patient, 10 to the minus 9th candela per meter squared, up to about 1,000. And then another new stimulator that we've released just this year called the color flash, we can go from 10 to the minus 3rd up to 10 to the plus 3 or 1,000. So typical thresholds, I won't go through all of these numbers, but basically this is a very powerful suite of tests where you can test a normal patient all the way to a patient that's light perception or even no light perception, and there's a range of about 1 billion to 1 visual acuity or visual sensation, across the range that we can test with the color dome. The tests themselves can be reported in candela seconds per meter squared, candela per meter squared, or decibels, and you'll see that in the presentation today. The basic test then for FST is reported in this sort of format. It's a very simple chart. Basically, you're reported a threshold for each eye or both eyes if both are tested at the same time, as well as a quality of the test. We use a Weibull function to calculate the threshold of the patient themselves. This has been proven over and over again to be a good mathematical technique for measuring the threshold of a patient. That is plotted then down to the bottom edge of the graph, and you can see the difference here between a normal vision patient and a low vision patient. So why use the test? Primary reason, and we'll talk a lot more about this as we listen to our guest speakers today. Low vision patients test the ability, the test is able to quantify a person's vision when all or nearly all of the other tests are unable to do so. So over and over again, people have reported getting high quality, repeatable measurements off of light perception subjects, and we've even had some reports in peer-reviewed journals of no light perception patients being tested. It's a proven and highly repeatable method. This is our method was used in the Spark Luxturna trial and the predecessor research for the Luxturna trials. There's over 50 peer-reviewed papers now, clinical use worldwide for over 15 years since it was initially developed in centers, including the two that our two speakers are from today. And then there are 5 high-quality peer-reviewed studies that have found the repeatability of Diagnosys's FST system to typically be in the range of plus or minus 0.3 log units of candela seconds per meter squared, or if you're reading in a decibel chart, that's plus or minus 3 decibels. Compared to visual fields or similar tests, it's a full field test, so this enables the best possible chance for a low vision patient to see the light. It's a fast test, typically 2 to 3 minutes, in some cases 1 to 2 minutes, so it's quite fast. That's after dark adapting. and it does not require fixation. Compared to electrophysiology measurements, the FST method has the ability to quantify vision levels 10 to 100 times lower than the best ERG methods, and sometimes even lower than that. Compared to other diagnostic tests, visual field tests, very similar concept, but again, designed for low vision patients. The SPARK trial highlighted the fact that FST is well correlated with the maze test or the multiluminance mobility test, as well as dark adaptometry, as I mentioned, is a similar type of test to our FST system. So that was a quick introduction. I will now turn things over to Dr. Aleman. Tomas?
Hi, good evening or good morning, depending where you are. And speaking to you from Philadelphia, from the University of Pennsylvania. I want to introduce you to two main topics in the use of full field psychophysics. One is the indication. So when do you need to think about using this type of testing, either in a research setting or in the clinic, as well as some of the basic limitations that we should all be aware. So the indications, I guess the simple kind of indication is to quantify vision, which is pretty much the indication to do an electroretinogram, or the indication to do a visual field. But in the case of full field psychophysics, we are typically using this technology when other tests are out of range or near the outer range, when they are informative, so that they are not providing you with some information of the visual attributes that you want to describe, or when they are unreliable for various reasons. Because of that, is a test that is used often in severe vision loss, patients with legal blindness, not necessarily total blindness. It's a test that is can be used in children, and I will talk about and show you a few examples in a minute. It serves as a complement of other measures of vision. That is, you don't have to think about the FST as your only measure, but it can be used together with other measures such as electroretinography, perimetry, to kind of provide more information about the visual function of the patient. And it can be used as a measure of safety or efficacy of the treatment, as Jeff has alluded to. So there is a long history of using full field stimuli to measure vision. Spans at least 4 decades. We at the University of Pennsylvania were confronted with the problem that is frequent in retinal degenerations when we were planning to do the initial gene therapy trials, and of that effort came this the initial paper where we actually tried to use the Humphrey perimeter to present a diffuse flash of light to patients with severe vision loss. So what is the situation that you're presenting in presented in the retinal degeneration clinic, or just in the ophthalmic clinic? That sometimes common measures such as the electroretinogram, which has a very long history of being precise and very quantitative, is no longer able to provide you with the measure of function, especially for rod-mediated function. So cone-mediated function by ERG can be measured with different approaches to basically when it's nearly undetectable. A rod function presents a little bit more of a complexity. In this graph that you see to the right, it's actually a group of patients that we tested initially with full field psychophysics, with full field sensitivity testing. And within this circle, you see dark bars, and the dark bars are patients that have visual fields that are limited to about 2 degrees or 2%, and with ERGs that are near the noise level or undetectable. So in those cases where you don't have an ERG or a visual field that is substantial, we were able to reliably not only quantify the vision of the patients, as you see here, the scale, vertical scales in dB with full field stimuli, but we were able to determine which photoreceptor was mediating the response, which are the little tick marks that you see here at the bottom. You are able to determine by using chromatic FSTs whether rods, cones, or both are mediating the responses. That is one of the main reasons then that we decided, we and others, Dr. Birch, who is in the lineup today to speak to you, modified basically an electroretinogram, mostly electroretinogram system to be able to do full field psychophysics with the diagnosis. And usually you have no problems in patients who have fields like this one here on the middle panel. You have a lot of sensitivity to measure. The problem is when you get to patients that is within 5 degrees or so of the center. In those patients, you can expect fixation problems, and you can also expect that in the near future, they may lose their function altogether. So in those situations, it's very important to think about the FST. It will save you the aggravation of not having measured something when you could, and not being able to compare down the road with the test and follow-up of these patients. So it's a very common scenario that we see all the time. The patient doesn't have to be 20 or 200. It can be a patient with 20 or 20 acuity, but very limited fields. In those cases, predict that things may get worse in the future and that you will actually lose the ability to measure their function with perimetry, for example. And that's an excellent situation to complement your testing with FSTs. Another scenario is this one here, which is the use of the FST in young patients. So testing children is not impossible. We can do visual field in patients that are as young as 5 years of age, but it's variable. Kids will, depending on their participation in the test, they may be terribly unreliable. And the FST can be modified to deliver, for example, to children by using binocular stimulation, by having the patients participate like in a game, in a video game, by having the parents of the patients participate in the testing. As Jeff mentioned, it's a relatively brief test that does not depend on fixation. So it's relatively simple psychophysically for children to perform the test. In this table here, for example, that comes from a paper that we recently published, the range of ages were from age 4 to age 17, and all of those kids, most of which had very severely affected fields and acuities, were able to perform the test reliably. One example of the utility in young kids is this. Here I'm showing an OCT from a patient with LCA that has very limited function, but by perimetry you're able to measure cone fields, which are very limited. They are very basically you only detect cone function near the center of the fovea. If we stop there and did not do FSTs, we would have missed something that is very important, and that is that we may have run out of range to measure rod function, and that's exactly what happened in most of the patients in this study. When you then went ahead and did the FSTs, all of the patients, instead of being just tiny islands of cone mediated function, were actually rod mediated in a situation that is somewhat unusual for early and severe retinal degenerations. And that was only possible with FST, given that most of these patients were totally out of range for detection of a stimulus, normal stimulus by conventional perimetry. Another is not okay. Another possibility that you have with FST is to relate the values of sensitivity that you determine with perimetry, and we know that it relates very well with perimetry and electroretinography. And you have also the possibility to do pupillometry, which in the newer versions of the diagnosis systems, you can actually do pupillometry as is shown here in the plot to your right, the threshold measured by FST, or the sensitivity losses measured by FST, relate very well with the sensitivity losses mediated by pupillometry. The traces on the left are not ERGs, they are actually pupillometry traces to increasing sensitivity, to increasing intensities of light in a patient compared to a normal subject. So it's a nice, it can be nicely complemented using the same instrument with objective measure of function beyond the retina. And as I mentioned before, it has been used effectively to define improvement in vision in the gene therapy trials for RPE65. Shown here are examples of FSTs before and after gene therapy in RPE65 LCA. This is not from the clinical trials. This is now with administration in clinic of the treatment. And as you can see, the top bars, we have them coded blue and red for the two stimulus colors that we use. The difference between the two colors provides you a measure of which photoreceptor is seeing the light. In this case, pre-treatment, most of the thresholds are driven by cone or both rods and cones, whereas after the treatment, all of a sudden your blue bars become really large. The increases in sensitivity measured by FST go from 100-fold to 40,000-fold, and the retina converts from a cone-mediated retina to a rod-mediated retina. So again, in a situation of severe vision loss, you have not only a measure of sensitivity, but a measure of photoreceptor participation, specific photoreceptor participation in the responses. So what are the limitations? Well, the main limitation is that it does not provide you with regional information, and that has to be taken into account when figuring out what the test means to you, as well as when we are delivering the test to our patients. This is the simple interpretation of the field of the FST values. You have a field that is extremely restricted to about 5 to 10 degrees of eccentricity. To the right is a plot of the sensitivity of the retina to a blue light. And in this case, you have an island, and the peak of the island corresponds to the FST. So there is no problem there. There is no confusion. Both FST and perimetric measures are beautifully correlated. There is, however, the possibility of a complex interpretation when you can have two areas within the retina of different sensitivity. And it's important for us to recognize that the FST is measuring the sensitivity of the retina, or the area of the retina with the best sensitivity. So in this case, for example, the FST is capturing the sensitivity of this island to the left, and is kind of ignoring what is happening to the center of the field. Why it's important to recognize that? One example is when you're measuring treatment. In this example to the left, you have the little island of function before treatment, and then after treatment, you have a new island that forms with sensitivity increase that goes from an absolute escotoma to plus 40 dBs. If you go by FST, however, your change in sensitivity would underestimate the treatment effect by a lot in this case. Of course, this is not what always happens, but it is a possibility, and you have to consider that. And likewise, you can have a treatment effect like this on the right panel, that if it's lost, then you can end up with a, for example, a single item of vision, and then you would be underestimated the loss of the efficacy of the treatment. So it is also important to recognize that the person that is delivering the test has to instruct the patient very accurately, because the sensitivities of the retina can be different. You know, in this example, again, using the same example, you can have an island in the periphery with different sensitivity compared to the center. The patient has to be told to, for example, press independent of the location of the stimulus, because for the patient, it may look like the stimulus is coming from the center, if the sensitivity is maximal at the center, or in the periphery, if it's coming from the periphery. They should be instructed to ignore the location. And this is important because many of our patients are actually well trained in performing perimetry, and when first faced to do a task such as the FST, they tend to follow the instructions of, having done perimetry for, 20, 30 years, and they get really confused. In this example, a patient, for example, is given to press when the light is just presenting straight ahead, and they will give you here to the left in the table values of sensitivity estimates that are 10 dBs less than if the patient is instructed to press for the dimmest light that they can see. So that is very important for technicians or for ourselves. And I recommend that if you are initiated into full field psychophysics, you dilate one day, dilate your pupils, darken out, and do the test to realize that this is very different from just doing the plain perimetry that we all love. And I think with this, I will pass the baton to Dr. Birch, who's going to take over.
Okay. With no further to do, I will continue. So thank you, Tomas. That was great. So as Tomas nicely described, the FST was originally developed in the Jacobson sedation Aleman lab to measure thresholds in severely affected patients. And so the initial papers were taking patients that had very severe retinal degenerations and just finding some way to quantify the residual vision. And it was really kind of leading up to the LCA trials. These were kind of preliminary data that were needed to support the clinical trials moving forward. Oops. Sorry about that. So in 2007, Alejandro Roman and colleagues were the first to use the Diagnosis color dome to present the full field stimuli. And the original FST that they developed was really modeled on the Humphrey, on the staircase. The Humphrey uses a 4-2 staircase to define threshold. So it was very comparable to the paradigm used in static perimetry. To make this available to others, they worked with Diagnosis. Diagnosis released its software for the dome, and it was a little different. The Diagnosis FST software used a Weibull function, a 2-parameter Weibull function, to do a staircase that was somewhat similar to the original staircase. But there are some key differences, and Richard Robson, I guess, did a lot of the programming. And basically, the Weibull function takes into account the false positives, false negatives, and the accuracy of the patient. So it's different, and it was important to determine whether the two measures really gave you the same kind of thing. So one of the first things we did getting into this was to compare the two measurements on the same dome. So we had the Diagnosis software and we had the Aleman software, and we looked at the two, and what we found was a very close relationship between the two. So if you look at this middle graph here, the DFST values that we got out of a large group of patients with severe disease were very similar to the FST measurements. We also looked at repeat variability, and this is something that has been looked at a few times now. And what we found was very comparable to others, that the 95% confidence range in this Bland-Altman type analysis, the 95% confidence range goes from -3 to 3 decibels. So it's a highly repeatable test. Those kind of values are very comparable to the values that we find in other forms of perimetry. So at this point, having sort of validated this a little bit and looked at the repeatability, we started measuring FST in a variety of patients. So like Tomas, we measured it in patients that had poor acuity, poor fixation in kids that were too young to really do visual fields. But we also found it very useful in patients with almost non-detectable or non-detectable ERGs. So, you know, as we follow patients, we usually have them come back every 2 or 3 years. Often the patient's rod ERG in particular is non-detectable. And in a situation like that, we do the FST in the same session. So they're dark adapted already, we do the FST, and then from that point on, we use the FST to follow the patients over time. So we found it very useful, and we're actually getting together a large number of patients that we've been following just to look at the follow-up data. But this was an early attempt to do that. This is just 12 patients with no detectable rod ERG, and they had FSTs on 2 visits at least 12 months apart. So the average threshold on the first visit was 4 log units, so 40 decibels above mean normal. So they're quite advanced patients. What we found is that most of the patients that were tested at greater than 25 months intervals showed a significant, greater than 3 decibel increase in threshold. So, you know, as long as it's been a couple of years, we're finding that most of these patients show progression in the FST, and there's really nothing else that we can use to follow this. So that's been particularly useful. So low vision patients and advanced patients are great. It's very important to have the FST available. We've recently begun looking at how useful the FST would be in less advanced patients. And we incorporated the FST into the RUSH-2A natural history study. Now, this is a study that's being supported and performed by the FFB Clinical Consortium, and it's 16 clinical sites in North America and Europe. And we have 127 participants. These are all 8-year-olds with at least 2 disease-causing USH2A sequence variants. And as far as clinical diagnosis, USH2A causes both recessive retinitis pigmentosa, and in this case, that was about 60% of the participants. I'm sorry, about 40% of the participants. And it also causes USH2A with hearing loss, and that's about 60% of the participants. So we have a variety of outcome measures. These are measures that all the centers could perform, and they included visual fields, acuity, OCT, FAF, autofluorescence, ERG, visual function questionnaires, and FST. So that's the only measure we have of rod function in this study is the FST. Most of the patients don't have detectable rod ERGs. So it was important to get the FST in, because otherwise we would have nothing that allowed us to look at rod function in this large cohort. And so we're able to follow that over time. And we've just looked at the data from the initial year, the first year of testing. And this is kind of a summary of all the patients. Virtually all of them had an FST. And what we found is that there's a very, significant difference between the USH2A and the recessive RP patients. So these are all pretty much the same mutations, but in one case it's causing a syndrome, in the other cases it's causing just RP. But nevertheless, there's a 0.001 difference in the sensitivity of on the FST. So it's able to discriminate between these 2 groups of patients with USH2A mutations. There was also a significant relationship with the duration of disease. So we asked all the patients when they first had symptoms, when they first were diagnosed. But as far as duration of disease, those that had duration of less than 10 years had much lower thresholds on the FST than patients who had a reported duration of disease of greater than 20 years. So it's looking promising, just based on the early data. And to sort of drill down into this a bit more, we've been looking at the the white data and the blue and the red data. So in the study, we're dark adapting the patients, and then we're determining thresholds to white, and then to the blue and the red stimuli to determine whether the patients retain rod-mediated function. And this is just kind of shows you the rationale for a normal subject. The blue is a little bit more sensitive. Patients are a little more sensitive to the blue than they are to the white, and they're much less sensitive to the red. And there's about a 25 dB difference on average between the blue and the red in sensitivity. So in patients, as they initially affected, patients that have not had the disease for very long still show some rod-mediated thresholds, and the difference between the blue and the red is still maybe 20 dB or so. As the disease progresses, the rods become more affected. All of a sudden, the red stimulus is picked up by the cones. It doesn't take very much loss of rods for the red to be cone mediated. And then we have a little bit of extra sensitivity to the blue in this particular case. And this is kind of a mixed situation where the blue is being determined by the rods, and the red is being determined by the cones. And then finally, as the patients become more advanced, they all start falling pretty much on top of each other, because the red and the blue are matched with the cones. So if they are giving you the same thresholds, then basically telling you this, all that's left in the retina is cones. So we looked at the relationship between the blue minus red difference, the difference in decibels, and the FST white threshold. And what you can see, it's kind of a revealing figure, because it shows that most of the patients have a blue-red difference of greater than 10. So most of them lie in this area. And the open symbols show the patients with autosomal recessive RP. And you can see that most of the recessive RP patients fall in this category where the red-blue difference is greater than 15 or 20. The solid symbols here show the USH2A patients, and they tend to fall in the cone-only area, where the difference is less than 10. And then we can also look at the number of years since diagnosis. And in this case, the blue and the red are the patients that have been that have had the disease the least amount of time, and the black is the patients that have had it for the longest. So it shows that it falls out pretty well with duration and with the diagnosis of the patient. The other thing it shows is that there's kind of a cutoff here about minus 30 dB. And it's saying basically that if you have a white threshold that's lower than minus 30, so between minus 30 and minus 65 or so, then you can be sure that that white threshold is determined by rods. And these patients all have this red-blue difference of 15 or greater. On the other hand, the majority of patients who have thresholds that are within less than minus 30 dB have thresholds that are determined by cones. So it's a useful finding, because now we can say that if someone's elevated by maybe 35, is a 35 dB elevation from normal, we could say that at that point we know that thresholds are going to be determined by cones at that point and higher. So just one other point we've been looking at. We also have incorporated into the study visual fields, dark adaptive visual fields on a Medmont perimeter, and not all sites have it, but I think about 70 patients have been tested on FST and the dark adapted fields. And we looked at the relationship, and it's really powerful. There's about a 0.85 correlation between the most sensitive value on the dark adaptive visual field and the FST. So we always say that the FST measures the most sensitive region of the field, but this actually proves it, because we can actually take the most sensitive point on the dark adaptive field and correlate that with the FST, and it's a very strong correlation. So it's providing something that's very useful. Just to finish up, just quickly, just a potential novel application. We've been talking to several groups, companies that are looking at visual cycle modulators, especially in Stargardt disease. As you know, Stargardt involves ABCA4 mutations, and they affect the way that retinol is cleared from the photoreceptors, and there's a much larger deposit of substance that needs to be degraded in the RPE, and it overloads the RPE. So one possibility for treating or slowing down Stargardt disease is to block the visual cycle, to block the RPE 65, and prevent the visual cycle from producing the rod debris. So you're sort of sacrificing the rods to save the cones is kind of the idea. Right now, it shows down here, this is the blocking the RPE component of the visual cycle, and it basically slows the whole cycle down. And this can actually be measured, and rod ERG has been used to monitor the degree of rod inhibition in patients in these trials. And this just shows an example of prior to dosing, if you get a nice rod ERG, and then if you bleach the retina, it knocks out the rod ERG, and then you can follow it coming back over 30 minutes or so. And after dosing, this is after 2 weeks of this amixistat, what you get is a dose-dependent inhibition of recovery. So the patients getting the highest dose, 10 milligrams, show very little recovery in 30 minutes. And the companies are using this kind of data to titrate the dosage to the patient. And it sort of occurs to me that the rod ERG maybe, it's very nice way to do it, but not everybody has it. FST might be a much more practical way to evaluate the inhibition from these drugs. So I think that's an application that's sort of waiting to be utilized. And I think Jeff's already kind of referred to the possibility of using it in AMD, early age-related macular degeneration. There are definitely defects in dark adaptation there. This might be useful for that. So I think at that point, I better stop and let you take some questions.
Excellent. Thank you very much, David and Tomas. Before you, both excellent presentations again from two of the most experienced clinics and clinicians and doctors in working with Diagnosys's FST system. So thank you again. We will open things up for questions, and Natalie will run this portion of our session. Natalie, the floor is yours.
You're on mute.
Every time. All right, thank you. First question we have in chat, is the FST used in animal models? And the answer to that is no, we don't currently have a behavioral setup for animals with the FST, although I do know some people have been working on sort of analogous projects, but no, right now the FST is a human-only module. Next question, how do regulatory agencies view the FST as an outcome measure? And I know that's something that we've all been talking about. So I'm going to punt that one to Dave Birch and Jeff Farmer and Tom Aleman.
Sure.
They've talked about it more than I have.
Well, I think with any of these tests, the FDA wants to know how it relates to the patient's everyday life. And so I think one of the keys to establishing the FST as a valid outcome measure is going to be continuing to look at its relationship to, for example, the maze, or its relationships to measures or instruments of PROs, patient-reported outcomes. You know, so I think we all know that the FST helps to discriminate degrees of blindness, and we have to convince the FDA that it's doing that too. And I think, you know, we've come a long way. Tomas has shown some great data along those lines. But I think, you know, ultimately the FDA has to know that somebody with a better FST has a better visual lifestyle.
Question for Jeff. You had mentioned an eye patch setup to measure regional FST loss, the eye mask. How does this work for low vision patients with wandering eye movement?
So that's a trade-off, certainly. The eye mask is newer. As I mentioned, it was developed, as I showed on the chart, with Dr. Mike Marmer at Stanford. Mike was interested in using it for dark optometry, but it applies for FST as well. So one important consideration when you consider using an eye mask to restrict the field of view and not be full field, say it could be a 50-degree field of view test through the eye mask, is the ability for that patient to be able to fixate well enough to be able to look through that 50-degree field of view hole in the eye mask rather than full field. Most important question. To the extent they can't, then it's not an appropriate test for them. To the extent that they can, then this is one technique to help refine the analysis, similar to what Tomas mentioned in his last couple of charts, of looking at full field sensitivity versus central sensitivity using an FST technique. So IMAS certainly can help in that way, and it's starting to be implemented in research by a couple of our customers.
Could I just add too that that's particularly relevant to AMD, because if you're going to do dark optometry or FST in those patients, it's to me it'd be kind of unlikely that you would see a big defect if you tested the whole retina. You know, it's going to be much more obvious if you limit it to the central 50 degrees or so.
That's a very good point, David. And we do have in the IMAS the ability to test different spots. We're basically blocking a portion of what you can see in the color dome. And so for customers that are using the eye mask for AMD patients, it's a rather small spot, similar to a Goldman-Weakers type spot of just a few degrees. Great point.
So another question. Sometimes patients with severe IRD note photopsia is another noise that's hard to interpret when a dim stimulus is also present in a FST flash. Do you have a strategy to coach subjects to detect the FST rather than and help them discriminate from those photopsias and no other sort of visual noise?
It's a great question, and it's a problem, you know, no question. I mean, it's a problem for all types of visual fields in patients. I mean, we do have spontaneous flashes, and sometimes they're difficult to distinguish from the real thing. I think the fact that there's a tone, you know, that you're looking at something that's temporally linked to a sound, I think that helps. So. But I don't really have any good answers to that. We just try to show the patients what the flash looks like. You know, we always, even when they're dark adapted, we show them something that's a little bit above their threshold, just so they could see what it looks like before we do the test.
Tomas, any thoughts on this point?
Yeah, we usually, I agree 100% with David. So the trick usually that works is to present a super threshold stimulus, you know, a log unit or so above the predicted threshold for FST, so that they see, as David mentioned, what it's supposed to look like, which doesn't look like a photopsia. So photopsias, no matter how severe they are, the patients know they're photopsias. And They are very different from the actual firing of the FST. And I think that's the best you can do. People have tried to calculate the level of desensitization from a photopsia with fancy algorithms. I wouldn't recommend that. It will confuse the patient in many cases unless you have time to conduct research into that specifically. I would just present the suprathreshold stimulus and familiarize them with the test.
I'll just quickly mention, it's a great question, two very insightful sets of points from David and Tomas. And our software automatically does do some of these techniques that David and Tomas have brought to bear here in checking for errors with no flashes, but on the flip side, showing very relatively bright flashes relative to the threshold, even late in the test, when our software has a pretty good idea where the threshold is at that point. Our algorithms are still moving the flashes of light up well above the threshold. And there's a couple of reasons for that, but one of the most important one is what David and Tomas just mentioned, to help remind the patient what a flash of light looks like, which even for normal people is important just by definition when we're testing near a threshold.
So another question from chat, are DTL electrodes the only electrodes used in FST testing? And also, can this be done on infants or young children? So for this test, you don't actually need electrodes. The input is not electrical, it's just a button box. So you do need a cooperative patient. This does require a degree of performance. So no for infants. For young children, I actually recently asked Tomas this myself, what the youngest patient he tests is. And it's very child dependent, is what I was told. I think you said as young as 4, if you can get away with it.
Yeah.
Let's have Tomas talk. Yeah, about that.
Depends on the kid, but 3, 4 can do it for sure. The tricks that we have played is make it a game. Have the kid sometime, sitting comfortably on mom's or dad's lap and bring the gansel towards them, and instead of doing it uniocularly, you can do it binocularly. You can actually let the parent press the button. I'm sorry, I'm clearing my throat. Pressing the buttons for the patient. One beauty of the system is that it has you have visualization of the pupils and the face of the patient basically, and in some ways you can also have that as evidence that the patient is actually paying attention and looking at things. So a kid that is kind of distracted and all of a sudden tries to search for a light when the flash is presented is an evidence that they saw the light. It's like a preferential looking type of thing. It's not a preferential looking test, but you have a lot of things that you can play with to be able to do the test in children.
Another question. It's been emphasized that the FST measures the most sensitive area of the visual field. This suggests that spatial summation isn't very important for the test. Any thoughts on the smallest area that would generate a normal threshold?
I don't know if David wants to talk about that. Yeah.
Go ahead.
Go ahead, yeah.
Oh, I'm offering you the opportunity.
So we don't know the actual. So spatial summation has to be taking part here. I'm not aware that like what has been done with perimetry has been done with FST. We certainly have not explored a relationship, but spatial summation, I'm sure it takes place as it does for any other psychophysical measure of vision. How small, as Jeff has mentioned before, even patients with no light perception, where we assume that actually the perception may be occurring from non-classical photoreceptors, the FST type of system is able to produce thresholds. So even in that type of situation, where even the summation of photoreceptor is out of the question, and you're dealing with proximal retina, the FST is able to still produce something. Now, if the question is whether it has been modeled, I'm not aware it has been modeled. I know that it relates beautifully to the psychophysical measures of rod and cone function by perimetry. So by extrapolation, we can say that yes, it must be dependent on the integration and the spatial summation.
Very good.
That's the last question so far out of the chat box, unless anybody else has got something that they haven't put in yet.
I have,
close to the top.
as we're waiting to see if there are any, let me toss out one question to David and Tomas that we haven't talked about yet. Can you each touch on the amount of time you feel appropriate to dark adapt for a dark adapted FST patient, and to the extent it varies by patient types, just comment on that?
Well, we do 30 minutes, and we feel like we're sort of comparing it to dark adapted fields or to raw ERGs. We want to be kind of consistent. So we do 30 minutes, but that's 30 minutes of patching, or in a dark room. We then take the patient and set them up in the test and stuff. So I think, by the time they actually begin the testing, it's probably more like 40 minutes. But I think 30 is safe. It doesn't change much beyond that.
Very good. That's excellent. And I think we have quite a few people on the webinar who participate in clinical trials with our FST system. And what we always re-emphasize is each of the trials have their own protocols, and follow what's in the protocol. So everybody's doing the same thing.
Right.
I have one other, Natalie, unless there's a question from the group.
No.
Nope. David and Tomas, could maybe each of you just touch on, outside of the clinical research, the trials that you've participated in for many years, just the patient types. You both mentioned that you use FST clinically with patients, especially when you get a flat ERG, but can you talk about what types of patients those are?
So it's not only, so I guess what I was trying to convey is that this is not a test for blind people. Sure, legally blind patients who are impossible to test otherwise will be the perfect candidate for this situation, but it's a test that will help you in patients with severe loss. So one example that David and I have worked recently are patients with choroideremia, and choroideremia has preservation of the central cone function with excellent acuities until very late in the disease, but they often become unreliable for normal perimetric estimates. Their ERGs are usually for rods undetectable from very early in life. So it is one group of patients, for example, that exemplify that you should not wait for someone to be an LP to be considering FST, and to remember that like in choroideremia, you can have a patient that today maybe 20 or 20 and able to do, I don't know, a 10-2 visual field, but in 5 years down the road, you may have no comparison. And by establishing a measure of vision ahead of time, you will be able to continue to plot the natural history of the dysfunction over time. And hopefully down the road, if there is a treatment that becomes available, then you have a measure to compare against. Otherwise, you're basically left to the last test that you did, and that's kind of the wrong approach to this type of diseases.
Excellent.
Yeah,
David, any additional thoughts?
Yeah, just that we began in severely affected patients. We thought of it as a sort of in-game test, and more recently, I've been more and more it's been more and more useful in less advanced patients. And this, Rush 2A trial is a good example. It's providing a lot of information that we weren't getting from any other device. So I think it's, useful along the whole continuum of severity.
Excellent. Thank you both. Natalie, we're a couple minutes over. Unless there's anything else super important from the chat bar, we can head to the finish line.
It looks like that's it. We did have a question about whether or not we would be making a copy of the presentation available. We will be sending out those slides that we showed today, along with a recording of this webinar. So that will go out. If you received an email about this, then you'll get it from there. Otherwise, if you found out about this another way through social media or similar, feel free to send us an email, mail@DiagnosysLLC, sales@DiagnosysLLC, any of our email addresses, and we will send that out to you. So that we can certainly do. Darla, I can see your email. I will send you one directly. So yes.
Let me take the opportunity that, I'm sorry, Natalie, anything else?
Nope. And I was going to say, and that's, I think, all that I have. So
I'll just close out then by thanking both Tomas and David again. Tomas is a little bit under the weather today, so 2 gold stars for Tomas, and very much appreciated for both of you to take the time to prepare and join us on the webinar and share your experiences with us. Very much appreciated. Thank you.
I see a lot of friends on the attendee list, and I only wish I could be in person. It would be nice, wouldn't it?
Someday, every day we're getting closer, right?
Getting closer, yeah.
Excellent. Well, thank you again, both. Everyone have a fantastic day or evening, depending on where in the world you are. And thank you very much for joining us. Have a great day.
Thank you.
thanks very much.